There we go. All right. We are now going to introduce our next speaker, Dr. Santhi Ganesh, who is Associate Professor of Internal Medicine and Human Genetics at the University of Michigan. And Dr. Ganesh has been one of our international leaders in understanding the genetics of fibromuscular dysplasia. She's also a member of the Medical Advisory Board of the FMDSA, and she will be speaking on current understanding of the genetic basis of FMD and what clinicians and patients need to know. Santhi, Dr. Ganesh, welcome.
Thank you so much. Thank you for that introduction, and thank you to Pam Mays, the FMDSA, all the organizers for the invitation to speak today and share some of our really exciting progress in the area of genetics research for FMD. Just by way of disclosures, nothing financial, but there they are. And I'm going to talk a bit about what we've been up to as far as genetics research for really some number of years now, and focus on the questions of what have we learned about the genetics of FMD through our ongoing research? What are these genetic findings teaching us about the biology of FMD, and what is clinically relevant now? So going back a little over a decade ago, we formulated a number of questions about the genetics of FMD. Chiefly, what is the architecture of FMD? So what that means is, are there possibly many genes involved that kind of each somewhat subtly contribute to a disease? And that's known as a complex disease with more common genetic variants. And we'll talk a little bit more about what that means. Can there be interactions between these genes? Can there be interactions between these genes and the environment? Can there be other changes to the DNA, which are called epigenetic effects? Or is FMD really a Mendelian disease where there are rare variants with a very strong effect on a protein that's encoded by the gene that then doesn't work properly and causes a more severe disease? And in those sorts of diseases, there are specific familial patterns. For FMD in some families, it's been supposed that it's autosomal dominant, which means you need one copy of a change to show disease, but with what we call incomplete penetrance, meaning not everybody with a variant will show the disease necessarily. So those are some of the complexities in terms of how we might think about the approach. And really our question was, what genes underlie FMD? And the last point here about how do we achieve precision medicine has to do with how do we identify people who might be at higher risk for certain events. I think that's a really important question in the field. So in terms of familial disease in FMD, anywhere from 7 to 11 or so % of individuals with FMD are reporting family members with FMD. And I want to walk through a couple of considerations that are important to understand the relevance of our findings and our approaches to the research, where when we think about genetic variants, they have a certain frequency in the population. Very rare variants that damage the protein that results from the gene tend to have what's called a high effect. And on the other end is more common variants that kind of contribute subtly to disease. And what we see here is the Mendelian form, the single gene type of disorder. Up at the top here, low frequency, high impact. And we identify these typically through pedigree studies and sequencing of DNA, as opposed to more complex genetics, common genetic variants, which we identified through an approach called Genome-wide association analysis, which I'll try to explain it very briefly. And these tend to be variants that change how the genes function. And then in the middle, kind of an intermediate category of maybe more severe forms of FMD, we tend to apply genome sequencing to identify both rare and lower frequency variants, potentially variants that are acting as what we call modifiers. And I'll talk a little bit about that as well. Just to give an example of the kinds of challenges that I think we're up against in the genetics research, is that this is an example of multifocal FMD and a renal angiogram affecting both of the renal arteries. Here is the histology under a microscope. We see that there is thickening and thinning of this blood vessel in that arterial media that Dr. Kim nicely explained in her talk just before. And here's an intimal fibroplasia, unifocal stenosis type of FMD, which has a very different histology, and underlying biology is what we infer that when we see that the histology is different with more of a growth in that intimal space. So the question that comes up here is, when we're doing our analyses, are we really comparing apples to apples, or are we sometimes mixing apples and oranges? And that affects our study approach and our results. The other complexity is that when we look at pedigrees, where here's an individual affected with FMD, here's a sister with FMD, that we also need to think about, how do we know who is not affected by disease? And in some older generations, sometimes we don't have access to angiographic imaging to get a diagnosis, but we might hear things like the grandmother had a stroke at age 35 or very early onset hypertension. And then how do we think about additional arterial findings, like if there was a spontaneous coronary artery dissection with the relationship to FMD? We know that the manifestations of FMD can vary within an individual. They can be variable across families as well. So this is just to give you an idea of what we're working with here. And again, a familial pattern suggests a single gene disorder. A non-familial pattern is what we call sporadic and has some of these considerations of whether it's single genes or multiple genes. And then there can be genes that are linked to multiple diseases in a condition called pleiotropy. And here what I'm showing are two newer identified genetic loci that are associated with FMD, SCAD, cervical artery dissection at two specific loci associated with these genes here, the factor 1 gene, which I think we're going to hear more about later this afternoon from Dr. Kovacic, and the LRP1 gene, with varying odds ratios for association with these diseases, but all in the same direction, raising the risk. Really interestingly, there are other associations of these same genes that are coming out of our genetic studies. And I'll talk a bit about this, but migraine headache, blood pressure for the LRP1 locus, abdominal aortic aneurysm, and an inverse association, meaning that the risk factors for SCAD and FMD seem to lower the risk for atherosclerotic coronary artery disease at this factor 1 locus and at others. Very interesting. There are several genes that have been statistically associated. This slide is summarizing what has come out of statistical genetic analyses. There are many other genes that have been described to have single variants that are not included here. And we won't have time to review all of these findings, but I'd like to walk through a few key examples and really summarize what the major progress has been. So first, starting with common diseases, common manifestations, complex variants with common genetic variants, multifocal FMD and SCAD. We have had the pleasure of working closely with Dr. Boatia Naji and really many other investigators in the field. Really, many of the people we're hearing from today have contributed samples and genetic data from individuals so that we could pool our data into a meta-analysis and find novel genetic factors, which I'll describe briefly. Similarly, for spontaneous coronary artery dissection, there's been some progress with genome-wide association analysis for this disease as well. So just very briefly to describe what is a genome-wide scan or a genome-wide association analysis. We take our research samples here for FMD. We need to have control samples, so individuals who do not have FMD, and we are accessing something called the Michigan Genomics Initiative, where we have over 60,000 patients from our electronic health record data and with genetic data that are available to us for research. And we can combine our sequencing and further analysis. We can match many controls to every case by age and sex and ancestry using principal components of the data. Just to give you the sense of what that is, it's really trying to cleanly match so that you're comparing as similar profiles and really try to limit the findings that you have to what is specific for FMD. And then across millions of markers across the entire genome, we can look for association of specific variants with the disease of interest. And what this does is it identifies specific segments of the DNA, the genes that are within those segments, and we can use that data to develop what's called polygenic risk scores. And we'll talk a little bit about that. So in this international GWAS meta-analysis, I think something that's really been an absolute pleasure and a real foundation for the success of the work so far has been the highly collaborative nature of the investigators in this field, how everyone has come together, pooled together samples across multiple centers in the US and in Europe. with us at Michigan, combining with Dr. Gornick and her biorepository, but as well from the Mayo Clinic, Mount Sinai, from the group in France and Paris, our colleagues who have a trans-European consortium, and the Polish studies. And by putting everything together in terms of the genotyping data and conducting the analyses in each cohort, then combining it, we were able to find several loci that are associated with FMD for the first time. Factor 1 was previously described a number of years ago in a smaller genome-wide association study, and now we have multiple hits that are showing up. Here, looking at all FMD as well as restricting the analysis to multifocal FMD, I think points out one important point, which is that when we look at the odds ratio, meaning for each of these individual regions that was associated with FMD, despite cutting down the sample size by about 20% here, we see that the odds ratio is actually higher, which gets at that point of, are we comparing apples and apples or apples and oranges sometimes? And I think really paying attention to the pattern of disease and the subgroups that make best clinical sense is yielding good results for us. This is very technically complex, so I won't describe it in too much detail, but essentially to summarize how we arrive at specific genes after associating a location in the genome, we can access tissue repositories that have hundreds of individuals assayed, where we look at not the DNA, but the RNA that's expressed. And the RNA is what eventually encodes for the proteins that are made from the genes. Those RNA molecules can be assayed in large-scale ways that look at the sequencing information and give us the basic information of what gene is expressed in what tissue. And it gives us, for all of the genes across the genome, what is happening. We can integrate that with our genetic data. And really the takeaway for this is that we can prioritize very strongly certain genes out of our genome-wide scan. And this is really an important stopgap, because once you've done your genome-wide scan, you need to know what will be the gene that you need to follow with biological studies. So we're very pleased that we were able to prioritize several genes here. In terms of the genetic structure of FMD through the GWAS, correlation analysis, looking at blood pressure, migraine, cervical artery dissection, and then atherosclerotic forms of CAD and MI, coronary artery disease, and myocardial infarction or heart attacks. What we see is that the genetics of FMD is positively correlated with hypertension, systolic and diastolic blood pressure, and pulse pressure, which is not surprising. FMD, one of the major manifestations is hypertension. Interestingly, there's a positive correlation of the genetics of FMD with the genetics of migraine headache, suggesting that there's shared underlying genetic predisposition and biology. Same for cervical artery dissection, although the effect might be a little bit attenuated when we adjust for blood pressure, systolic blood pressure. And interestingly, when we look at that inverse relationship of FMD and atherosclerotic forms of coronary heart disease, we see that after conditioning for blood pressure, this inverse relationship is actually quite apparent and quite strong. So we've done the same for SCAD. In our group, we've collaborated with Dr. Saw and the CAN-SCAD registry, along with the University of Michigan MGI program, and analyzed the cases described here against the number of controls, conducted discovery replication analyses, pooled all of the data together, and identified 3 strongly associated genetic loci for SCAD. And we did some of the same things that we did for FMD, which I'll show you, but we also tried to extend these findings a bit to say, what can we learn about SCAD that occurs in the context of FMD? Can we look into general samples through resources called the UK Biobank, which has hundreds of thousands of individuals with their electronic health record data linked to genetic data, where we can ask kind of unbiased questions about the genetic structure of SCAD and what we see. And I'll walk through what that is. And we also went into the Million Veterans Program, where again, hundreds of thousands of individuals could be accessed to really explore this association with other forms of myocardial infarction or heart attack. And what we see here, again, doing very similar approaches as that RNA analysis I described for FMD, strongly prioritized these 3 genes for SCAD, we did work to show that this is actually expressed in that middle layer of the coronary artery that Dr. Kim had explained. And there are the specific cells of that layer of the artery, the vascular smooth muscle cells, that are producing this particular gene, Adams-like 4. And just as kind of hint as to where we're really putting some effort now, we looked at the difference in gene expression in arterial tissue according to whether these were women or men. And these are subtle but statistically significant and possibly biologically important differences in the expression of some of these genes according to sex. So I think this is a very interesting molecular kind of avenue to explore as we're thinking about these genes and how are they different in individuals with SCAD. When we went into this UK Biobank resource, we can ask the question, when we take all of the risk alleles for SCAD and we simply say, what else is it associated with? The main thing that popped out was migraine headache, very positive association there. And again, an inverse association. This is showing the p-value. So how statistically significant is it? But actually the direction of that association is an inverse. So the risk factors for SCAD were protective against heart attack and myocardial infarction due to plaque rupture. And these effects are consistent across men and women, which is notable. So just switching over now to rare variants, Mendelian type disease patterns. One example that I'd like to highlight is a new finding in a COL5A1 gene. And what we found were 4 individuals who had a specific genetic variant that had a very similar pattern of arterial involvement with neck vessel tortuosity, fibromuscular dysplasia, multifocal appearing type in multiple vessels, celiac artery aneurysm and dissection, as well as iliac artery aneurysm and dissection. There were a host of other findings. The dissections included multiple other arteries, like the coronary artery, for example, in one individual. But this variant was very interesting because what it's doing is it's causing a glycine, the amino acid, in the protein structure of this collagen molecule COL5A1 to be substituted. And glycine molecules tend to be very important for collagens. And when these are altered, the normal structure here where the collagen fibrils come together nicely and form a very well-formed bundle of collagen actually cannot occur normally. And so this gene, COL5A1, has been associated with something called classical Ehlers-Danlos syndrome, where there's joint hypermobility. There can be skin findings of hyperextensibility, so stretchy skin, and abnormal scarring. Very rarely, arterial dissections have been described, but never really a recurrent variant. And this variant is so rare that it was not observed in over 100,000 individuals with no disease in a reference database. But what we did was a really detailed ancestry analysis going back many, many generations. So these 4 individuals were not thought to be related. Our initial genetic data confirmed that. But when we really did some close-up analysis, what we found is that the chunk of DNA in which this variant is occurring is shared between these 4 individuals who, again, we had thought based on our typical ancestry analysis were not closely related to one another. And what this suggests is something called a founder effect, that a variant arose several generations ago and could be inherited on this piece of DNA, that then everybody who got that piece of DNA may have that variant. And so that's what this is showing. It's called a phylogenetic tree, showing where this variant is. And that to find a variant that has not been described before, a completely new variant clustering in this way and showing this effect is really very noteworthy. And what the suggestion is that's important to know clinically is that there may be other individuals in the population who have this particular variant. And that impacts our thoughts when it comes to screening. And this is a clinically actionable genetic finding. I'll talk a little bit about the clinical implications towards the end. But just to say that this is the kind of finding where it's damaging enough to the protein and it's causing a pattern of disease that we need to think about this when we see a very typical pattern, again, of the neck vessel tortuosity, a lot of arterial dissections, plus fibromuscular dysplasia lesions that have a multifocal kind of appearance. So this is not something we should have everybody tested for. Again, I really think it should be restricted to this individuals with this pattern with a very heavy burden of disease. And then we have talked about this study in prior meetings where a number of years ago, we looked at just under 50 individuals from the National Institute of Aging in a study on connective tissue disorders. And there were individuals with multifocal FMD in this cohort, 47. There were clinical features that we see in other diseases where the TGF-beta pathway is effective. And so we looked at TGF-beta expression in the plasma and secreted from dermal fibroblasts, so skin biopsy samples. We grew the cells and looked at what was actually being secreted off the cells. And TGF-beta 1 and 2 was elevated in patients with FMD in the plasma and on the dermal fibroblast cultures. So what this raised was a question of why do we see this? What is this really about? Is it telling us that this is a TGF-beta disorder? Is it a genetic disorder, the way that mutations in this pathway can cause aortic aneurysms and dissections in a syndrome called Loeys-Dietz syndrome? And there have been 2 series reported. One was 102 patients last year reported with a severe multifocal FMD phenotype, and 1 individual with a TGF-beta 3 variant reported for Loeys-Dietz syndrome. The other individuals did not have a variant, and there was no enrichment for those variants for FMD. So by not seeing an enrichment, it tells us this is not something that is likely affecting many patients with FMD. It might be rarely found. In a clinical series of FMD, this is by Dr. Gornick's group, actually, and there were 2 individuals with TGF-beta receptor 1 variants. And again, these are variants that can be associated with Loeys-Dietz syndrome. And so sort of what I have been watching the evolving data from our group and other groups, the implications are that really variants in this pathway, these TGF-beta genes, are observed rarely in a very small minority of patients to date, but not in the vast majority of patients with FMD. And we really need to continue to think about, why do we see elevated TGF-beta in the blood and from the skin fibroblasts? Is it a really causal finding? Is it just kind of a bystander, a secondary thing that we have found? We still don't have a grasp on this, but should have data that will help us to understand this in the coming years. And then finally, just about this intermediate category, where we're looking here at variants that have an intermediate effect. One question we thought would be interesting and timely to propose is, among those with FMD, do the identified genetic factors that I've described influence the risk of arterial dissections? And this would be considered a modifier effect, where you have a disease and then another variant kind of adds to this and then predisposes to dissections. That was the question. There are 2 studies, and Dr. Kim showed this same slide, so I won't belabor this except to say there's a lot of similarity between FMD and SCAD in terms of the demographic patterns and some of the overlaps. And so we asked the question, if we take that SCAD polygenic risk score and look at individuals with FMD in a cohort of a little over 400 individuals, and actually only 28 cases of SCAD, so really we thought this would be a very underpowered analysis. What we saw was that there is an association of these genetic variants with the occurrence of SCAD in individuals who already have FMD. So we saw that there's a lot of overlap in the genetic contributions from common variants to FMD and to SCAD. And what this is telling us, in my interpretation, when we think about the question of, as Dr. Kim described, sisters versus cousins, how closely related are these 2 diseases? From the genetic perspective, this suggests that they are partly overlapping. We see the same genetic loci for some of the involved genetic factors, but that we can find something that is differentiating individuals with FMD is intriguing, and I think important for helping us to understand that they're not one and the same genetically, but that there are some differences that are probably important to continue to pursue. And then thinking about the rare and low frequency types of variants, we looked at the collagen 5A1 gene. We had exome sequencing in 265 or so individuals, 264 individuals. And we found 7 individuals who had genetic variants that met certain criteria that are a bit technical But, essentially when we do predictions using bioinformatic methods, they're predicted to damage the protein greatly. And the frequency in the reference database called NOMAD is very low frequency or rare. So if a variant met these criteria, which 7 individuals had variants meeting that criteria, there was an association with dissection occurrence, and more specifically in the visceral arteries. So 43% of individuals with these collagen 5A1 variants had dissections, and versus 3.5% in the general cohort. None of these individuals had a diagnosis of connective tissue disorder, interestingly. And I think when we look at what we have in terms of histology, this is one of our G514S variants. We see that nicely organized elastin in a control normal artery, really broken down in an FMD artery. And what we have here is pooling in this collagen, the blue stain here, that is abnormal in FMD. So this we treat as preliminary evidence supporting that there are genetic variants that may have modifier effects, and we need more research. So just to touch on the question, do these findings have clinical utility? The rare variant that we described, the phenotype is actually quite reminiscent of vascular Ehlers-Danlos syndrome. So we have, in the publication, we described how we might think about this variant, and we've listed that here, thinking about certain kinds of activity restrictions to avoid isometric strain and blood pressure spikes, counseling to know what the signs of a dissection are. There's no medical therapy that is specific for this, and surveillance imaging that might mirror what we think about with vascular EDS. The polygenic risk markers are not ready for clinical application. We advise family members with vascular symptoms to seek evaluation for FMD already, and to engage cardiovascular genetics and genetic counselors in trying to sort through this. So very briefly, I think we might be getting close to the end of the time. I see Heather Gornick has joined. That we're gaining new insights into the biology and the genetic risks. We have several challenges. There's this issue of apples to apples and apples versus orange comparisons. Familial risks are somewhat uncertain, and we need really large sample sizes to really get at all of the genetic architecture. But we have many opportunities here to apply really cutting edge genetic methods. Our multi-center international collaborations are really pushing this field forward. We hope to clarify phenotypic subgroups, achieve precision health goals, specifically risk prediction, and really think about the biology. And in terms of our study at Michigan, I'll just put this slide up, and we are enrolling individuals with manifestations of FMD, aneurysms, dissections, family members, conducting all sorts of analyses ourselves, and looking to collaborate very broadly internationally to make the most of this data. So with that, I'll just put up my acknowledgement slide and say a lot of this work was led by trainees in the lab, excellent collaborators at the University of Michigan, really an extensive list of very important collaborators without whom this work would not have been possible. I'd like to thank clinical colleagues who refer patients, and really the patients and their family members who participated in the studies, and put in their time and effort as well. We have been funded by many different sources, and appreciate the support of the FMDSA and referring patients, and contact information and where to follow for new updates is below. Thank you.
Dr. Ganesh, thank you very much. A lot of rich information there. We are just going to have time for one question, and then we'll take other questions in our panel this afternoon. And very briefly, someone is in your clinic who has FMD. What should they tell their children, their grandchildren? What should they discuss with their doctors or pediatricians as of now?
Right. So as of now, if we have a suspicion that there is a very severe type of manifestation where we need genetic testing, that may guide what we tell patients. So we typically will say, let's see what the genetic testing shows, and if there's something there. But for most individuals, that actually is not the case. We're not testing the majority of individuals, and even when we do, it's most often that we do not find a genetic variant in our current testing. And in that case, really for individuals with symptoms that suggest a vascular kind of insufficiency, And that list of symptoms can be quite broad, to make sure that they talk with their physician and tell their physician that they have a family member who has FMD, and to think about really imaging as the first examination, discuss what those symptoms are, and consider imaging.
All right. All right. Well, Santhi, I hope you're able to join us for our panel this afternoon. We're going to have a good 45 minutes for lots and lots of questions. So again, hold those questions for this afternoon. And Dr. Ganesh, thank you so very much.
Thank you.